Hello, my name is Rupachandra. I'll be handling 21BTC101T Biochemistry for you. To begin with, Unit 1, Introduction to Biochemistry. The topics to be learnt under this is Introduction, Chemical Bonds, pH, Buffers, Carbohydrates, Proteins, Lipids and Nucleic Acids. So, Introduction to Biochemistry. So, all of you know that biochemistry is the study of structure, organization and function of all living matter. So, to understand about biochemistry, so your biochemical reactions are very important for all the diverse forms of life. So, molecular evolution of biology in 20th century has become the common language of all the biological sciences. So, the topics to be addressed by the biochemists. So, various biochemists have given the biochemistry definitions where they have mentioned the definition of biochemistry in different terms, where they have said that biochemistry is the chemical structure. It deals with the chemical structure of all the components of living matter. And certain biochemists have also identified that biochemistry involves the interactions of all the components that give rise to supramolecular structures, multicellular tissues and organisms. And biochemistry is also found to be dealt with extraction of energy by the living matter from the surroundings in order to maintain alive. And also biochemistry deals with storage and transmission of information for the living matter. And biochemistry involves the chemical changes that accompany the reproduction, aging and death of all the cells and organisms. And about biochemistry, the very important concept, if you know about biochemistry, you want to know about biochemistry, is it mainly involves the control of chemical reactions. Regulation of chemical reactions you can study in biochemistry. And to analyze about the major events or the historical events of biochemistry, so if you see the first starting discovery which was made in biochemistry is to mention is about the nucleic acids. So Mischer isolated nucleic acids in 1869, and further about the glycolytic pathway was also discovered in 1925, and to extend this, citric acid cycle was discovered by Krebs in 1937, and followed by that we have the discovery of the double helical model of DNA, a very super discovery by Watson and Crick. So these are the very important or significant discovery of biochemistry. And also if you see the other discoveries about biochemistry about 100 years, you can see the unveiling of genetic code, then 3D structure of hemoglobin, and also MacLeod and McCarthy have identified the genetic transformation process, and urease was discovered and analyzed that it was in crystalline nature. Then Buchner has identified the fermentation reaction in the cell extracts, where he identified that sugar was converted to ethanol. And Wohler have also identified urea, and he found that urea is synthesized from ammonium cyanate in the labs. Now further, to analyze the biochemical reactions, first you should know what are the biochemical molecules or biological macromolecules which are involved in biochemistry. So all the four major classes of biomolecules follow the same principle, which is called the central dogma of genetic transformation. So the biomolecules which are involved in biochemistry, the four major macromolecules are proteins, carbohydrates, lipids and nucleic acids. So all these four major macromolecules is what we are going to study in biochemistry. Now biochemistry is also found to be a subject which is interrelated with other life science subjects. To mention that biochemistry is also related to organic chemistry, which will tell us about the properties of the biomolecules, and biochemistry is also related to biophysics. So biophysics, all of you know, it is a subject which will deal with the structure and techniques which are related to the physics, and biochemistry is also related to another subject called medical research. So medical research involves all the medical pathways. We will be understanding all the metabolic pathways involved in the medical field. So that is also where biochemistry is related to medical research. Coming to nutrition, biochemistry is also involved in nutrition, where nutrition aspects are involved in biochemical pathways, where biochemistry is related to all the nutritional aspects of your dietary requirements of every day in your life. Then coming to the other fields where biochemistry plays an important role, where to start up with we have microbiology. Microbiology is an important subject which deals with all the metabolic pathways in microorganisms, which is also a subject related to biochemistry. And physiology, where we understand all the physiological processes in living organisms, is also related to biochemistry. Cell biology, which describes the biochemical division of all the cells, related to biochemistry. and genetics, where we understand all the mechanism of action of particular gene which is taking place, analysis with your biochemical identity. Now we'll first start with the first topic, chemical bonds. So all of you know macromolecules, they interact with each other by certain chemical bonds. To start with, first we'll analyze what are covalent bonds or non-covalent bonds. Okay, so covalent bonds are first the bond which is formed by the sharing of a pair of electrons between the adjacent atoms. And if you see here, you have paired electrons and unpaired electrons. So unpaired valence electrons, where you have atom 1 and atom 2, where these two unpaired electrons, when they are shared, they are found to form a covalent bond. So the covalent bond is formed by the sharing of electrons, which is seen in an atom. Now if you see here, there are also non-covalent bonds which are found in the macromolecules. The non-covalent bonds examples are electrostatic interaction or electrostatic bonds, hydrogen bonds, van der Waals forces. So all these are non-covalent interactions which are seen in macromolecules. Now we'll see what are non-covalent interactions. So non-covalent interactions between macromolecules are very important in all the living organisms. So if you see here non-covalent bonds, the first one, there are three types of non-covalent bonds: electrostatic bonds, hydrogen bonds, and van der Waals forces. So all these three covalent bonds are involved in the biochemical reactions of all your cells. So if you see here, the basic definition of an atom. So the basic unit of matter is going to be atom, and all of you know what atom is made up of. So atom is made up of protons, neutrons, and electrons. So protons are positively charged, as you all know about it. Neutrons, they are neutral, they are present in the center of the atom, and electrons are always negative, they are present outside the nucleus. Now coming to what are chemical bonds. So these chemical bonds are the ones which are going to be holding your atoms together firmly. So these atoms are held together strongly by chemical bonds. So these bond formation is formed by electrons which are going to surround the molecules, which are called as valence electrons. Now we'll see what is the mechanism of this covalent bond. So if you see here, covalent bond is formed by when the electrons are shared between the atoms. So they are also called as the strongest bonds in their chemical bond. So these covalent bonds, they are found to be 2 or more in number in a molecule. Now next we will see about ionic bonds. Now ionic bond is formed when one or more electrons are transferred from one atom to the other. So if you see an atom, when it loses an electron, you call it as a positively charged. Okay, when an atom, if you say it gains an electron, you say it is a negative charge. And positive or negatively charged atoms, they are together called as ions. So all the ionic bonds are not found to be very strong. Some would be weak and some would be strong. So ionic bonding, if you see, they are found to be one of the important bonds which are seen in all the macromolecules. Now coming to the next non-covalent bond, which is going to be van der Waals forces. So these van der Waals forces, they are found to be forces which are having the weak forces. So if you see here, sometimes certain atoms, they do have a non-covalent bond which are not found to be very strong. They are found to be very weak. For an example, if you see, they are seen in geckos. So these van der Waals forces are also seen in your lizards which are going to be climbing on the wall, where they are found to have only van der Waals interactions because they have weak interactions. So these weak interactions are very important for the molecules to dissociate and then to be held together strongly. So the van der Waals forces are found to be weak bonds, and they are also found to be seen very widely in geckos, where they are the climbing lizards. So these forces help the geckos for climbing ability. They help them for the ability for them to climb over the walls. So that is why we call them that these forces are van der Waals forces. And coming to the hydrogen bonding, so the basic hydrogen bond, all of you know, it is present in water molecule. So water is said to be a polar molecule where it has the hydrogen and oxygen atoms. So if you see, oxygen is negatively charged and hydrogen is positively charged. So the hydrogen bonds are found to be weak and they are classified under non-covalent interactions. Coming to the pH. So next topic we are going to see is about the pH. So all of you know the definition of pH. pH is the unit of measure which will tell us about the acidity or alkalinity of a solution. So it is identified between the scale from 0 to 14. So all of you know, if the concentration of H plus is higher than the OH minus, then you call the given solution to be acidic. And if the OH concentration is higher, then H plus material is found to be basic. So the neutral pH, where both H plus and OH minus ions are found to be equal, you call it as neutral, or the pH 7. Now the pH is found to be derived with an equation. pH is equal to minus log H plus. Now to understand the basic derivations of acid and base concepts, so a scientist called Lowry and Bronsted have identified a concept for the derivation of acid and base. So acids, they are called as proton donor, and bases, they are called as proton acceptor. So a proton is going to be always a hydrogen atom which has lost its electron. So according to Lowry concept, the acid is defined as a proton donor, and according to Lowry, base is defined as a proton acceptor. So if you see the reaction here, a base reacts with an acid to form a conjugate acid and a conjugate base. Now coming to the Henderson-Hasselbalch equation, the acid dissociation constant is given by HA is equal to H plus A minus, where Ka is the dissociation constant, and H plus and A minus divided by HA gives you the total Henderson-Hasselbalch equation, where Ka is defined as the dissociation constant.